Hello and very warm welcome. Dear students, today I will be talking about the types of female gametophyte or embryo sac in angiosperms. The plant life cycle is characterized by alternation of generation between a diploid sporophyte and a haploid gametophyte. The sporophyte functions to produce spores which then develop into gametophytes. Differentiated gametophytes in turn produce either the male gametes or female gametes. In contrast to lower plant species in which the gametophyte is the dominant free living generation, gametophytes of angiosperms are smaller and less complex than the sporophyte and are formed within specialized organs of the flower. In angiosperms, the male gametophyte or the pollen grains develop within the anthers and the female gametophytes or the megametophytes or embryo sacs develop within the ovule. Strasburger in 1879 was the first person to describe the female gametophyte in angiosperms. The process of development of female gametophyte can be divided into two stages called megasporogenesis and megagametogenesis. In general, during megasporogenesis, the megasporocyte undergoes meiosis and 4 megaspore nuclei are produced. Subsequent mitotic divisions, nuclear migration and cytokinesis during megagametogenesis produce the mature embryo sac. Considerable diversity in the pattern of embryo sac development is found among plant species. However, in most flowering plants, the embryo sac has a characteristic organization called the polygonum type, containing 7 cells of 4 distinct cell types. Depending on the number of megaspore nuclei taking part in the development, the female gametophytes of angiosperms may be classified into 3 main types called the monosporic, bisporic and tetrasporic embryo sacs. Each type of embryo sac has more than one subtype named after the genus in which it was described first. Figure shows a small representative sample of each of the modes of embryo sac development that have been observed. Now, let us discuss the monosporic type of embryo sac. In this type, only 1 out of the 4 megaspores take part in the development of embryo sac. All the nuclei in such an embryo sac are genetically identical because they are derived through mitosis of the single nucleus. There are 2 types of monosporic embryo sacs. One is the polygonum type. The embryo sac in the polygonum type is formed by the clausal megaspore of the tetrad and is 8 nucleate. The mature embryo sac comprises a 3-celled egg apparatus, 3 antipodals and a binucleate central cell. This monosporic 8 nucleate embryo sac formed by 3 divisions of the functioning megaspore occurs in more than 70% of the angiosperms. The enlargement of the megaspore is always accompanied by increased vacuolation, one large vacuole usually appearing on either side of the nucleus in the direction of the long axis of the cell. After the first division has taken place, the 2 daughter nuclei move apart to opposite poles. Most of the cytoplasm is aggregated around them and the rest forms a thin peripheral layer, the center being occupied by a large vacuole. The next division gives rise to a 4 nucleate stage, which is followed by the 8 nucleate stage comprising a micropylar and a clausal quartet. Of the 8 nuclei arising in this manner, 3 at the micropylar end give rise to egg and 2 synergids. 3 at the clausal end give rise to antipodal cells, 2 and the remaining 2, one from each pole, fuse in the center to form a secondary nucleus. Occasionally, embryo sacs are found with less than the normal quota of 8 nuclei. This is usually because of an early degradation of the antipodals, which obscures the true nature of embryo sac. Even when antipodals are present, they are sometimes overlooked because of their being situated in the narrow callosal end of the embryo sac, which is seen only in median sections. The second type of the monosporic embryo sac is Oinothera type. The organization of mature embryo sac in the Oinothera type is an egg apparatus and a uninucleate central cell. Gates in 1908 found that in Oinothera lamarckiana, the embryo sac is usually formed by the micropylar megaspore of the tetrad, which undergoes only 2 nuclear divisions instead of the usual 3 occurring in polygonum type of embryo sac. In this way, 4 nuclei are produced which organize into 2 synergids, the egg and a single polar nucleus. Since the third division is omitted and all the nuclei are situated in the micropylar part of the developing embryo sac, there is neither a lower polar nucleus nor any antipodal cells. Modowilski in 1908 independently studied species of Oinothera, Epilobium and Circaia and confirmed the observations of Gates in all essential respects. These 2 investigations were soon followed by several others, and this mode of development known as Oinothera type has been found to be a characteristic and constant feature of the entire family Onagraceae, having been demonstrated in more than 16 genera. The only exception is Trapa, which has an 8 nucleate embryo sac of the polygonum type, but this genus, as most systematists now agree, is best assigned to a separate family, the Hydrocariaceae or Trapaceae. A noteworthy feature in the development of Oinothera type of embryo sac is the concurrent growth of more than one cell of the tetrad. Eventually, it is the micropylar megaspore which functions, but sometimes it may be the callosal and occasionally both grow simultaneously, forming twin megaspore embryo sacs. Now, let us discuss bisporic embryo sac. In this type, 2 megaspore nuclei take part in embryo sac formation. Here the first meiotic division is followed by wall formation, so that a dyad is formed. Only one of the dyad cells undergoes the second meiotic division, whereas the other one degenerates. In the functional dyad, cell wall formation does not occur after the second division, and both the megaspore nuclei contribute to the formation of embryo sac. Each megaspore nucleus undergoes 2 mitotic divisions, forming 8 nuclei. The final organization of the embryo sac is similar to the polygonum type. Bisporic embryo sac is again of 2 types. First, the Allium type. The embryo sac in the Allium type is derived from the callosal dyad. A bisporic embryo sac was first described in Allium fistulosum by Strasburger in 1879 and has since been confirmed in several species of the genus. The megaspore mother cell divides to form 2 dyad cells, of which the upper is much smaller and soon degenerates. The nucleus of the lower divides to form 2 and then 8 nuclei, which give rise to an embryo sac with the usual organization. The second type of bisporic embryo sac is Endymion type. The embryo sac is formed by micropylar dyad. This type was first described in Endymion hispanicus. In this type, megaspore mother cell undergoes meiosis I, forming a dyad. The micropylar dyad cell is functional while the classal dyad cell degenerates. Its nucleus divides to form 2, 4 and finally 8 nucleate embryo sac comprising egg, 2 synergids, 2 polars and 3 antipodals. A different type of development has been observed in Amoebium alatum of the family Compositae. Here the first meiotic division is not followed by wall formation and the dyad cell is represented by a single binucleate cell. Both the nuclei undergo the second meiotic division simultaneously and the daughter nuclei take up a 2+2 position at the telophase and their persistent spindles form a joint structure stretching from one pair of the nuclei to the other. Cell plate formation is initiated across the equator of the united spindles, which then extends to the lateral walls, so that each of the cell of the dyad is binucleate. A spatial situation occurs in Schisandra chinensis. In some of the ovules, the development of female gametophyte is bisporic, but the final organization of the embryo sac is as seen in Oinothera. This is the only authentic report of a bisporic 4-nucleate embryo sac. Now, let us discuss the most common tetrasporic embryo sacs. In this type of embryo sac, both the meiotic divisions are not accompanied by cell plate formation, hence all the 4 haploid nuclei at the end of meiosis are enclosed in a common cytoplasm, forming a cynomegaspore. As all the megaspore nuclei take part in formation of embryo sac, a tetrasporic embryo sac is more heterogeneous than a bisporic embryo sac because the 4 products of meiosis involved in its formation are genetically different. Much variability has been observed in the nuclear behavior of the tetrasporic embryo sacs. Based on the number of post-meiotic mitosis, the number of groups of nuclei in the mature gametophyte and the presence or absence of nuclear fusion during the development, Maheshwari recognized 7 principal types of tetrasporic embryo sacs, namely Adoxa, Peperomia, Pinia, Drusa, Fritillaria, Plumbagella and Plumbago. The arrangement of the 4 nuclei in the cynomegaspore before the beginning of post-meiotic mitosis is of 3 types. First, 2+2 arrangement. 2 nuclei at the micropylar end and 2 at the classal end, for example, Adoxa type. Second, 1+1+1+1 arrangement. 1 nucleus at the micropylar end, 1 at the classal end and 2 placed laterally, 1 on each side. For example, Pinia type, Plumbago type, Peperomia type. Third is the 1+3 arrangement. 1 nucleus at the micropylar end and 3 at the classal end, for example, Drusa type, Fritillaria type, Plumbagella type. Depending on whether nuclear fusion occurs or not, number of post-meiotic mitosis in the cynomegaspore and final organization of embryo sac, tetrasporic embryo sacs are of many types. Now, the first is the non-nuclear fusion, where the non-nuclear fusion occurs. First is Adoxa type. It was described for the first time by Johnson in 1979-1980 in Adoxa moschatellina. There are only 5 genera in which this type of embryo sac development is more or less regular feature: Adoxa, Sambucus and some species of Erythromium, Tulipa and Ulmus. In this type of embryo sac development, the nuclei of the cynomegaspore take up 2+2 arrangement. All the nuclei of the cynomegaspore undergo just 1 more division to form an 8-nucleate embryo sac. The mature female gametophyte has normal egg apparatus, 3 antipodals and 2 polar nuclei. An interesting variation was reported in some species of Tulipa. In Tulipa sylvestris, vacuolation frequently commences at the megaspore mother cell stage, and all the 4 megaspore nuclei gather at the micropylar end of the cell, where they divide to give rise to a group of 6 cells, one of which is an egg and 2 nuclei. Tulipa tetraphylla show almost a similar type of development. After the meiotic divisions are over, 3 nuclei go to the micropylar pole and 1 to the causal pole due to the formation of vacuole. All of them divide again, so that there are 6 daughter nuclei in the upper part of the embryo and 2 in the lower. Cell plate laid down later, resulting in the formation of 5 cells at the micropylar end. One of these is egg and 1 cell at the causal end, leaving 2 free nuclei in the center. Since this particular mode of development occurs only in Erystemones section of genus Tulipa, it is known as Erystemones form of Adoxa type. Dear students, till now we have studied the various types of embryo sacs or female gametophyte, which were mainly of 3 types: the mono, bi or tetrasporic embryo sacs. The monosporic embryo sacs were further divided into 2 types, the bisporic were further divided into 2 types, and the tetrasporic embryo sacs were further divided into 7 major types. We have reached Adoxa type. Today we will be discussing Peperomia, Pinia, Drusa, Fritillaria, Plumbagella and Plumbago. Apart from them, there are some minor type variations within the normal type of embryo sacs. Now let us discuss Peperomia type. Campbell in 1899 and Johnson in 1900 reported that in Peperomia pellucida there are 2 mitotic divisions in the 4 nuclei of the cynomagaspore after taking up tetrapolar arrangement and result in a 16 nucleate megagametophyte. According to Johnson, in the final organization at the micropylar end form the egg and a synergid. 8 fuse to form the secondary nucleus and the remaining 6 are cut off at the periphery of the embryo sac. Campbell also observed a similar type of embryo sac development, and according to him the egg apparatus is invariably 3-celled. Approximately 8 nucleate form the secondary nucleus and the remaining 4 to 6 nuclei are cut off as antipodal cells. Fagerlint in 1939 made a detailed study on Peperomia pellucida. He found that the outline of the cynomagaspore may be more or less spherical or pear-shaped, and in the later case there is slight protuberance at the micropylar end of the cynomagaspore shows 3-celled egg apparatus, 8 polar nuclei and 6 peripheral cells, and the embryo sac derived from the pear-shaped cynomagaspore. Johnson in 1914 reported a different type of embryo sac development in Peperomia hispidula at the 8 nucleate stage. 2 nuclei are seen at the micropylar end and 6 nuclei at the causal end. At the 16 nucleate stage, 4 lie at the micropylar end and 12 at the causal end. 2 nuclei of the micropylar group now form the egg and single synergid as in other species. But the remaining 2 nuclei of the micropylar group and all the remaining 12 secondary nuclei, and there are no antipodals. As this is different from Peperomia pellucida, it is recognized as a subset, namely Peperomia hispidula type. Murthy in 1959, who studied several species of Peperomia stated that in the mature embryo sac, the egg apparatus may be 2 or 3 celled, and there may be 6 to 9 polar nuclei and 4 to 6 peripheral or antipodal cells. Maheshwari in 1963 opined that in view of such variability, it was not necessary to maintain the two subtypes Peperomia hispidula and Peperomia pellucida types. Now, let us discuss Pannier type. Stephens in 1909 described this type of embryo sac development in 3 genera of Panniaceae, namely Pannia, Brachysiphon and Sarcocola. Here the nuclei of cynommegaspore take up tetrapolar arrangement and divide twice to form a 16 nucleate embryo sac. The 16 nuclei are arranged crosswise, 3 nuclei of each quarter later cut off as cells, while the fourth remains free and moves to the center. There are thus 4 triads and 4 polar nuclei. Generally, the central cell of the micropylar triad alone functions as the egg. The central cell of the other triads also simulate the egg. This type of embryo sac development is found characteristic of the family Panniaceae and Euphorbiaceae. Maheshwari and Johari in 1941 studied the embryo sac development in Acalypha indica, which is almost similar to Pannier type, but differs slightly in the organization of mature embryo sac. Here each peripheral group consists of 2 cells and 2 nuclei of each quarter remains free and migrate to the center and fuse to form the secondary nucleus. Thus, there are 4 groups of the 2 cells and at the periphery 8 free nuclei in the center in this type of variation. Now, let us discuss Plumbago type. Hopt in 1934 reported this type of embryo sac development in Plumbago capensis. The 4 nuclei of the cynommegaspore are arranged in a crosswise fashion. They undergo another division resulting in 8 free nuclei arranged in 4 pairs. A nucleus of the micropylar pair is now cut off to form the lenticular egg cell. Of the remaining 7 nuclei, 4, probably 1 member from each of the 4 pairs, undergo a slight increase in size and gradually approach one another and function as polar nuclei. The remaining 3 nuclei degenerate occasionally, 1, 2 and all 3 of them are cut off at the periphery to form cells which persist and assume an egg-like appearance. Synergids are entirely absent. The Plumbago type also occurs in Plumbago coccina and Plumbago scandens. This type was also found in 2 genera of Plumbaginaceae, namely Ceratostigma and Vogelia, and is not known outside this family. Now, let us discuss Drusa type. Hakanson in 1923 in Drusa opustifolia of Amblyophyrae recorded a 16 nucleate embryo sac of different nature. After the 2 meiotic divisions were over, the 4 nuclei of the cynommegaspore take up 1 plus 3 arrangement, and this is followed by 2 plus 6 arrangement and then 4 plus 12 nucleate stage. The 4 micropylar nuclei give rise to an egg apparatus and the upper polar nucleus, and the 12 classal nuclei to a lower polar nucleus and 11 antipodals. Later, this type of development was also noticed in Melotus japonicus, Crucianilla latifolia, Smilacina stellata, etc. Sometimes due to the failure of division of some classal nuclei at the 8 nucleate stage, there are sometimes fewer than 11 antipodals. The embryo sac of Mainthemum canadense also shows Drusa type, but is about 13% of the ovules. During the division following the 8 nucleate 2+6 stage, the callosal spindle fuse in pairs, resulting in 6 diploid nuclei instead of 12 haploid nuclei. Now, let us discuss Fritillaria type. This type was first described in Fritillaria, persica and Allium. The cynomagaspore in this type of embryo sac development takes up 1+3 arrangement. The 3 callosal nuclei are present very close to each other. During the next division, the micropylar nucleus divides normally, but the 3 callosal spindles fuse to form a single common spindle, so that at the close of the division there are 2 haploid nuclei at the micropylar end and 2 triploid nuclei at the callosal end. One more division occurs, resulting in 8 nuclei, of which the 4 callosal micropylar and are haploid. The mature embryo sac finally consists of an egg apparatus of which all the cells are haploid, the 3 triploid antipodals and a tetraploid secondary nucleus formed by the fusion of 2 polar nuclei, one haploid and the other triploid. The fusion of 3 callosal nuclei of the cynomagaspore during the division was first observed by Bambiconi in 1928, hence the fusion process is known as Bambiconi effect. Cooper in 1935 found the Bambiconi effect and the formation of Fritillaria type embryo sac development in the entire tribe Lilioidae and several other genera belonging to diverse families like Piper, Tamarix, Cornus, Rudbeckia, Gaillardia, Tulipa, Clintonia, etc. The fusion of the 3 callosal megaspore nuclei may take place when they are either in the prophase stage or in early metaphase. In the former case, the secondary 4 nucleate stage is processed by a secondary 4 nucleate 1, and the subsequent sequence then is as follows: megaspore mother cell, primary 2 nucleate stage, primary 4 nucleate stage, secondary 2 nucleate stage, secondary 4 nucleate, and lastly the 8 nucleate stage. Normally, all the 4 megaspore nuclei are of same size, but in some plants in the micropyle nucleus is larger than the rest, in which case the nuclei of the secondary 2 and 4 nucleate stage show no appreciable difference in size in spite of their triploid nature. Rarely the callosal nuclei are smaller than the micropylar, although they are triploid. Sometimes the basal nuclei of the secondary 4 nucleate stage fails to divide as a result of 6 to 7 nucleate gametophytes are formed in some species of Statica and Gagia. Depending on this, the mature gametophyte has either 1 or 2 antipodals. Now, let us discuss Plumbagella type. This is a reduced Fritillaria type of embryo sac and is reported only in Plumbagella micrantha. The 4 megaspore nuclei take up 1+3 arrangement. A large vacuole separates the 3 callosal nuclei from the micropylar. The 3 callosal nuclei eventually fuse to form a single triploid nucleus and results in secondary 2 nucleate stage. This is followed by a secondary 4 nucleate stage in which the 2 micropylar nuclei are haploid and the 2 callosal are triploid. There are no further divisions. The nucleus nearest the micropylar end organizes into egg. The triploid nucleus nearest the callosal end organizes into a single antipodal cell, and the remaining nuclei one haploid and the other triploid fuse to form a tetraploid secondary nucleus. This mode of development is similar to the Fritillaria type, and the only difference is that in Plumbagella, the development is arrested at the secondary 4 nucleate stage and the 4th division is omitted. Now, let us discuss aberrant and unclassified types of embryo sac. In addition to the 7 fairly distinct and well established types of tetrasporic embryo sacs, there are few which appear to be more or less isolated. Of this, the embryo sac development in Chrysanthemum cinerariefolium is special and interesting. The embryo sac development is sometimes somewhat peculiar in this taxon and was reported by Martinelli in 1939. The embryo sac is tetrasporic and the megaspore nuclei take up 1 plus 2 plus 1 arrangement, so that there is 1 nucleus at each pole and 2 nuclei in the middle. The 2 central nuclei become separated from the terminal nuclei by vacuoles and may either fuse to form a single diploid nucleus or may simply remain close to each other without undergoing any fusion. Based on the behavior of the 2 central nuclei, there are 2 variations within this. First variation is the 2 central nuclei fuse to form a diploid nucleus. After fusion, all the 3 nuclei divide twice to form the 3 groups of 4 nuclei each. From the micropylar quarter are produced the egg apparatus and the upper polar nucleus, all haploid. The clausal quarter gives rise to 4 antipodals, all of which are haploid. The central quarter is comprised of diploid nuclei. One of these functions as the lower polar nucleus and the remaining 3 are organized as additional antipodals. Occasionally, fewer than 12 nuclei are found either because of failure of some divisions at the clausal end or because the central diploid nucleus undergoes only 1 division instead of 2. Now, let us discuss the second variation. In the second of the 2 alternatives, that is the 2 central megaspore nuclei do not fuse, but only lie in contact with each other and function directly as the polar nuclei. The micropylar as well as the clausal megaspore nuclei divide twice, forming 4 nuclei at each pole. Thus, the embryo sac is 10 nucleate. All the 4 nuclei form the clausal quarter organized as antipodals. Of the micropylar quarter, 3 nuclei organize into egg apparatus, while the 4th one along with 2 central nuclei function as polar nuclei. However, there is no regularity in the behavior of clausal nuclei. Sometimes there is only 1 division or none, and based on that the mature female gametophyte is 9 or 7 nucleate. Occurrence of more than one type of embryo sac development in the same species of or even in the same individuals is common in some cases. Kapil and Prakash in 1966 in the Delosperma cooperi reported 14% ovules showing polygonum type of embryo sac, 18% Endymion type and 68% various tetrasporic types. Similarly, AB in 1971 recorded Allium 31%, Adoxa 53% and polygonum 16% type of embryo sacs in Orchid, Orchis patterns. But the interesting thing was that the mature embryo sac contained only 6 nuclei in all of them. In the genera like Chrysanthemum, Erigeron and Euphorbia, the development of megagametophyte may be mono, bi or tetrasporic. The environmental conditions to some extent can influence the type of embryo sac development in some cases. Hagelmist and Grazi in 1965 have shown that the temperature influences the type of embryo sac. For example, Ulmus glabra show Adoxa and Drusa type of embryo sac in addition to some rare types. According to them, low temperature 15 to 19 degree centigrade considerably enhanced the frequency of 8 nucleate adoxa type of embryo sacs. They explained that the temperature affects nuclear division more than the cell growth and vacuolation. At low temperature, the nuclear division is delayed in relation to cell growth, so that the central vacuole appears after meiosis 1, separating the two megaspore nuclei to the end of the megaspore embryo sac. By a single mitosis after meiosis 2, an 8 nucleate adoxa type of embryo sac is formed. At high temperature, the nuclei divide rapidly in relation to cell growth, so that the vacuolation appears only after the second meiotic division, separating a single nucleus at the micropylar end from 3 at the clausal end. By 2 more divisions, a 16 nucleate embryo sac of the Drusa type is formed. In capsicum, it was reported that the embryo sac formed in winter were bisporic and those formed in summer were of the monosporic type. Thus, the environmental factors like the temperature can influence the type of embryo sac development. With this, we conclude our today's topic. Thank you.